So genomes and genetics. Let's start. This is the third lecture topic. What we're going to do in here that just give you a brief overview of the structural part of the genomes and their genetics. And then the later part in the chapter, you will see that we will dive into each one of them in much detail. All right. So what I'm trying to do is, this is the first slide. So first, what we are seeing, that there is a virology breakthrough, that what we are seeing here. And this is a Hershey-Chase experiment, which you guys actually all learned or started to learn in high school, like DNA is the genetic material. And this actually came out in the 1940s. And then the mid-1950s, the nucleic acid, the DNA was already, the structure was solved in 1953, Watson and Crick. And these were some of the early evidence that there is DNA inside these viruses. And then Franklin and Conrad's work, they found that the tobacco mosaic viruses and many other viruses actually contain RNA. But there was a weird thing in there. There was a theory that has been pushed that for the viruses are like, say, bacteriophages are metabolic products of bacteria. That was pushed for a lot of time. And then a lot of people actually thought the protein may be the carrier of the genetic information to the progeny of these materials. So a lot of confusion in there. But then the Hershey-Chase, this is Alfred Hershey and Martha Chase in 1952, did an experiment, and it's called a kitchen blender experiment. And what you can see that if you just go to anyone from New York here, if you just go to Long Island, you go all the way to the end to the Cold Spring Harbor Laboratory, you will see that this setup is in the Cold Spring Harbor Laboratory anyway. So what they did here is that they took these bacteriophage, T4 bacteriophage, and the E. coli, mixed with them. But before mixing with them, they labeled the proteins with a radioactive sulfur, and they labeled the nucleic acid with 32P phosphorus. Then what happened, that once they labeled everything nicely, they mix these bacteriophage with the E. coli for a short period of time. So what will happen, these E. coli will absorb these bacteriophages on top of them, and they very quickly inject the DNA inside the bacteriophage. So after this short period of time, they took this bacteriophage and then they put it in a kitchen blender and blended it at a high speed. I mean, like, you don't want to do that radioactive material in a kitchen blender. Probably you will just have a radioactive aerosol, always, no safety, nothing. But once they did it, then they isolated the material that they blended in the blender, and they actually centrifuse it down and look at that what radioactivity they find in the pellet where you will have the E. coli bacteria. And what they found, that they actually found the DNA in the radioactive in the progeny phage. How it is? So basically, all these bacteriophage, after injecting the DNA, remained on the outer surface, and when you blended, they just all sheared off and they remained in the supernatant in the centrifuge. Whereas the E. coli, which where the DNA has been injected, that goes inside the E. coli, and part of that will be packaged into the progeny virions, because DNA replication is semi-conservative. Part of the strand will go into some of them, and so in the pellet, they actually found the radioactivity. And it was already proven, the classic experiment, that nucleic acid carries the genetic information. That's about it. So next, what we are going into, that the bigger surprise is the thousands of different virions, seemingly infinite complexity that what you have. But just remember this number, 7 days in a week, and there are 7 types of viral genome that is known to this. And the principle of this course will be basically to understand these 7 types of viral genomes and their strategies, and then how they actually work in these. Sometimes it can get really complicated. Here we are giving an overview of all the genomes, and then we will go into the detail of them later. So here, two key facts will make your life easier. And this is basically a recap. And all viruses on the planet also follow this rule, like all organisms. This is a recap, your central dogma, that how the information flow actually happens. So your central dogma in the molecular biology tells you DNA to RNA to protein. And that, again, we are reiterating that in the viruses with a little bit difference. So the virus need to make the mRNA, which we call as a transcription, and the viral genome must make mRNA that can be read by the host ribosomes. And the genome, which is packaged inside the box that we say, the viral genome, must make copies of its own. And don't worry about this. You will understand that when we talk about these slides in a little bit later. But this actually caused a little bit of problem. The problem is that the scientists actually fought about the naming of the transcription and replication. So the virus actually make the mRNAs to be read by the ribosomes, but the scientists actually, a group of scientists, not all, that's the reason that there are two ideas. One group of scientists actually argued that transcription means that you make RNA from DNA. And this RNA is not only mRNA, it is ribosomal RNA and transfer RNA as well. So if that is the case, telling viral transcription all the time, which is really the virus only makes the mRNA, will be wrong. You will be precise about telling that making of viral mRNA. But again, remember that we use the term making of the viral mRNA and transcription interchangeably, the same way we use analogically the anthropomorphic terms to tell about the viruses. We just cannot avoid that. So this is the idea. On the other hand, for the genome replication, there was not much controversy because your genome, no matter what, if it is RNA or DNA, the viral genome replicates that was similar to other organisms that is been found. And remember that the transcription is also from RNA to RNA from the RNA viruses. And that's the reason that other than transcription, we try to make the making of the mRNA from the genome rather than just telling it viral transcription. Now here, these are some of the homeworks that you need to work on your own. And I do not need to go into the detail of explaining about all of these as such. So just from your recap of molecular biology, this is needed, and a thorough understanding of this is absolutely required in this course. So the double-stranded RNA that you are familiar with, right here is a coding strand with 3 codons, and then you have a template strand. And there are some other names for this. The coding strand is also called the sense strand, and this is also called the antisense strand. And these are complementary. You already know that. So if you're writing a double-stranded DNA like this, the sequence will be like this. But if you have the same genetic information in a double-stranded RNA, only the T's will be changed to U's. Very simple. Now, when you make the mRNA from that, see that you have these coding strand right here. It is the exact same strand, and only you change the T's right here to U. So that's how it is. And there is a convention, and this is the huge concept that is coming in here. David Baltimore actually talked the coding strand right here as a plus sense RNA. Plus sense RNA, because these RNAs, which is like the viral mRNA, which comes from here, this is the code that is translatable immediately upon entering a cell and or after being produced by the ribosome. So that's why we call this viral mRNA as the plus sense mRNA. And this is the plastron RNA. It's exactly the same. The negative strand RNA, on the other hand, is 3' to 5', which is the complementary strand of the plus strand mRNA. And this is also found in some of the viruses. On the other hand, you can see the plus strand DNA is just only the top one, and the negative strand DNA is only the bottom one. Now, what you need to do it's not an assignment, but just on your own, you write the double-stranded DNA sequence. And just using these sequences, try to find out whether you can write all these different strands and how you reach to that strand. Remember that if we ask you that you have this negative strand RNA, how you are going to make the mRNA of that, this negative strand, first, you have to write the complementary plus strand, and that will be your mRNA. If you are given this plus strand RNA and asked to write the viral mRNA, how you will make it from this, you will have to make an intermediate of the negative strand, which is complementary to this. And then you need to make the complementary of this, which will make this, this viral mRNA. So that's pretty much the overview of that. That's how it is. It is very self-explanatory in that sense. So just work on that, just write it down, it will help. So here is also another overview of that whole thing, is that the viral genomes must make the mRNAs and replicate it. And if you have these, say, 4 genes in a viral genome, they make the mRNA synthesis, which we may sometimes call transcription. After being transcribed, they will make the viral protein or the proteins to make the box. And this particular area is the box. So on the, what happens, the genome, which is the full length of that, see that genes actually making individual, they are color-coded, individual mRNAs. That will make translated into the viral protein. And the genome is the full length copy of that. And that needs to be copied by replication. And that full length will be packaged. This is the word that we use, packaged inside or encapsulated inside the capsid or the coat of the virus. Or think about that, it is going inside the box. And these red marks are the sum of the intergenic sequences. It may or may not be present. And here it is not required. So the Baltimore scheme is a simple way to think about the virus. And this is David Baltimore, Nobel laureate. He was the president of MIT and is a very, very well-regarded molecular biologist and virologist overall. And he actually put this scheme to understand all the different 7 types of the viral genome. But he actually did not discover the 7th type. He actually came up with the 6th because the 7th type was discovered much, much later. So what he did, that he put the mRNA at the very middle of this scheme, because remember that all viral genome need to make the mRNA. And then he actually put all the other viruses around that and wrote the scheme that how these viral genomes can reach the mRNA. So you can easily see here, the plus RNA, we talked about that. The plus RNA need to make the minus RNA. On top of that minus RNA, you make the plus RNA. Double strand, on the other hand, let's start with the plus minus double-stranded DNA, because this is the easiest to understand for you guys from your classic organism molecular biology. You have the both plus minus strand. So all it does is that on the minus strand, which is the light blue in color, you just make the RNA by an RNA polymerase. That will be the plus sense of the mRNA. So this is the easiest one. If it is a plus RNA, it has to make first a minus DNA. From there, it makes a plus minus DNA. And then you can go to the mRNA as well. So all different strategies that you can see that there are 2 different strategies. And right here, when the RNA makes into the DNA, you can easily tell that what it is. RNA to DNA, this is your retrovirus, RNA to DNA, reverse transcription. Transcription is DNA to RNA that we talked about, but here when RNA is made into an intermediate DNA and the DNA makes the mRNA, this becomes a reverse transcription. On the other hand, you can see if you have a double-stranded RNA, you can make the mRNA. If you have a minus-strand RNA, just make the plus mRNA on top of that. That is easy. The one which was not, on the other hand, just let me quickly explain that you can have a plus or minus DNA. You just have to make the complementary strand. Once you get the duplex, you can get to the mRNA. The one which Baltimore missed, not missed because this virus was not discovered then, that there is a gapped RNA. And this gapped RNA may have a reverse transcript. It's very complex type of replication and transcription. And we are going to talk about that in detail as well. So spend some time to understand that. And we have several slides on this. So here that the 7 classes of the viral genome, once again, that all the different types that what you have. And this is the SS plus RNA with the DNA intermediate that you can easily see here. The gapped double-stranded DNA is this one right here. So all of these So again, just only one thing, that SS plus RNA with DNA intermediate. This is what it is. And this is the one that is found in the retroviruses. We're going to talk about that later. So here in the words, this is the Baltimore skip concept worksheet, and you can use this chart as a guide. So remember that in the notes I have written that use the slide number 6 right here, slide number 6 and then 8 to understand pretty much that how you can make the transcription, make the mRNA and the genome. That's basically the two. So genome replication, how you make the from genome to the copy of the genome, and how the genome makes the mRNA, and what are the intermediate steps. So if you just combine the 6 and 8 together and spend some time, you will be able to find it out. And it is just like a simple concept. And once you get it, you will never forget. You do not have to memorize anything from here once you get the concept of that. You need a complementary strand to make the strand back, that concept of molecular biology. All right. So a lot of different viral DNA, RNA genomes. And again, you do not have to memorize. This is just a pictorial reference that we will use throughout the course. And I have listed all of them in the notes, and I'm just pointing out a couple of different things in here, but you will be familiar with all these pictures throughout the course. So here you can see that this is a linear genome where the dark blue is the plus strand DNA, light blue is the minus strand DNA. On the other hand, you can see that if you are taking a double-stranded RNA, the bright green is the plus strand, olive green is the minus strand. And along with that, you can have segmented minus strands. Then you have a circular DNA right here, and there are covalently attached ends. You have a gapped DNA. You can have a single-stranded DNA. The UTR are the untranslated regions. Negative sense RNA. Ambisense RNA is interesting that one part is the plus and the other part is the minus. The sequences are in that way. Sometimes this ORI is the origin of replication, and this 5' dot that you are saying, that this is a 5' terminal phosphate that is attached in some of the viruses that you will be able to see those as well, found in DNA and RNA viruses both. Then there could be covalently attached proteins. Along with that, means that this is AOH means that these are poly-A tails. Poly-A tails are signature of mRNAs, and sometimes they are added 200, 300 poly-A tails at the end of the mRNA, and helps in the translation process. So these are some of the things that you are going to see. And sometimes in this one with the gap DNA, same example, that an RNA is covalently attached to one end and a protein is attached to one end as well. All right, so here, what is the function of the genome diversity? And what we actually do think that RNA actually evolved first in this world, because RNA do have self-replicating function, or it can catalyze its own synthesis. So, but there was a problem that we think, and again, there is no direct proof, that RNA genomes basically were much more unstable because of the nucleases, which degrade the RNA pretty quickly. And then at some times, probably, and again, we are using the term probably, we really do not know, that reverse transcriptase actually evolved where the RNA genome is converted into the DNA genome, which can be much, much bigger, much, much robust. And that's how the evolution actually went on. On the other hand, the RNA genomes also competed, and under the selection pressure, they also evolved period of time, and they never actually got deleted in the process of evolution. And there are something which called the viroids, and these viroids are just naked circles of RNA. They infect plant, cause pathogenicity, but they do not code for any protein. So these are some of the interesting observation. And one of the interesting thing that you can think that coronaviruses actually do contain some of the largest genome in between 29 to 34 kbs. You do not see any RNA genomes bigger than that, although you will see megabase genome in some of the giant viruses with a DNA genome. Now, the coronavirus actually do have a nuclease, which is a proofreading nuclease. That is not present in any other viruses known still in the RNA viruses. And this proofreading nuclease basically repair the DNA if there is an error. And so that's one of the problem in the sense some of the mutagenic drugs, which actually can cause some of these base pair changes, these are called the nucleoside analogs, which make errors or put these analogs inside the RNA when it's being made. And those RNAs will be non-functional. You cannot extend those RNAs. But it has been shown that these nucleases actually can quickly excise these nucleoside analogs from nucleotide when it goes inside the RNA. They can excise these out and repair it. So that's one of the problems in there. So here, this is also the important thing is that just a little bit of memorization in this chapter, that each of these virus families do have one, or here I just added one extra, names in there. Why we have these names? Because these are the classic examples of each of these classes, and we are going to refer to this all the time. So if you just remember this, and I believe me, there will be more names later, but I will not ask you to memorize any of these to understand the context. All right. So these are self-explanatory. So the next thing is that what information is encoded in the viral genome, and you can see that this is a completely self-explanatory. So the regulatory signal for replication of the viral genome, efficient expression of the genome, that means that, again, I use the word transcription to save space, but making of the mRNA and translation of the mRNA for protein synthesis, assembly and packaging of the genome. That means that once the genome is copied, it needed to be put into the box, and that is the assembly and packaging. And then regulation timing of the replication cycle, and modulation of host defenses. And most viruses need to at least, this is important, need to at least make one protein, at least one, excuse me, and which actually can modulate host defense. Without that, there will be no fruitful viral infection. The host defense mechanism can suppress most of the viral infections, and the mechanism to spread to other cells and host by different structures, which can bind to receptors or can fuse with the plasma membranes. All right, so what information is not encoded in the viral genome? Very important that no genes encoding the complete protein synthesis machinery is found. There is no ribosomal RNA or no ribosomal or translational proteins. Now, you have to be careful with that what we mean by no ribosomal RNA or ribosomal or translational protein means this is the structural component of the translational machinery. What happens that some of the DNA viruses even can make tRNAs. Now remember that tRNA is not the part of the ribosome, that's a transfer RNA. Amino acid tRNA synthetase, this is the enzyme which makes the tRNAs. So the viruses even can make that. On the other hand, they can make the translational initiation and elongation factor. Now remember that these are translational elongation or initiation factors which is required for the efficient start of the translation along with these structural proteins. But these are not the structural components of the translational machinery where there is no example of a single virus which can actually do that. So these are some of the things that is not encoded in the viral genome. And membrane biosynthesis, there is no such, and there is also no telomere or centromere found in the standard host chromosome in the viral genome. So here, these are, again, lots of names, and you do not have to memorize any of those. So what we are talking about, that if you just remember this, and I have underlined these things in your notes so that you know that what is important for your quiz and what is not. So here, just remember that the upper limit of the known viral genome. So approximately, say, 2.5 megabase. Megabase is this limit, about 2,500 protein. And interestingly, they are bigger than some of the bacterial genomes. Now, why this is important, remember that a bacteria is a free-living organism. It needs a huge amount of genome to perform metabolic function. So the Haemophilus influenzae actually does have a 1.8 megabase genome, which is a free-living. It causes problems or pathogenicity, but it is still free-living. It can live on its own. And so you can easily see that some of the viruses exceed a free-living organism genome size. On the other hand, this is Nasuia. This is an endosymbiont bacteria that is found in the insects. And this actually have less than 200 genomes, and they are about 112 base pair. Many of the viruses are even bigger than that. You can easily see that. This Nasuia is not free-living. It cannot live outside the cell, so it goes from cell to cell, and it's an endosymbiont bacteria from the insects. And here, interesting thing that you can see that all these mama virus, Mimi virus, Mumu virus, all these names, where these are all discovered from a French laboratory, and they gave these names, and it is kind of funny to find those. And you can easily you can just look at them about fun facts about those. All right. So on the other end, smallest known viral genome. We talked about the virides. These are just naked circles of RNA, no protein. Then the satellites are basically found as naked RNAs along with a viral infection, which actually may cause more pathogenicity, like a hepatitis delta viral genome. You can see right here is a length of 1700. 1700, but it just codes just only one protein right here. Some of the satellites are just naked RNAs. Some of the satellites may have just one protein, but extremely small base pair. So up to this right here, hepatitis delta satellite, we will not consider them as the true virus as such up to this, although it codes for a protein, but the satellite doesn't appear if you don't have a viral infection. So the smallest genome of the virus is actually this circovirus, about you can see 1759 base pair, 1700 base pair, just code for 2 genes. That's about it, 2 genes. So these are some of the overview of that. And again, you do not have to memorize these, just if you remember that what is a viroid, what is a satellite, and the kind of a lower limit of a virus. That's all you need from here. So the viral genomes actually differ from the host chromosome remarkably. The host genetic system is based on the DNA, and you know that RNA is a genetic material for the viruses as well. But many of the DNA viruses actually emulate the host. But however, almost all DNA genomes are not like the cell chromosome, and you have unexpected tricks. And what we understand that you can easily see that the genome of the eukaryotes particularly, you have the nucleosome structures, which is the association of the histones. And there is just only exception is the polyoma and the papilloma virus, which actually does pack the genome with the histones. The others actually don't have histones. On the other hand, many of the viruses actually cover the entire genome, particularly RNA viruses, with a protein coat that is also not seen in the host genome. And then what I did that in the, particularly in your notes, I just said that what are the different tricks that have been evolved, and you can just read those. Some of them may not make sense to you right now, and you do not have to memorize those. I'm not going to explain those as such here because we will study them later. And what the tricks are that from the small genome, how it can make many proteins. They can make polyproteins, they can make nested transcription, they can make subgenomic mRNAs, they can make overlapping genes. There are RNA can be edited, spliced, and lots of different tricks that I have listed in the notes have been evolved, and we will talk about them in detail later in the chapter. So, but just for a take-home message that the viral DNA genomes are very different from the host chromosomal structure or even prokaryotic chromosomal structure. So double-stranded genomes, you have a couple of viruses here: adenoviridi, herpes viridi, papillomaviridi, polyoма, and the pox viridi. And again, as I said, that few representative groups, you can keep them in the back of your mind. And I just exactly wrote this in the notes, but memorization is not necessary other than those representative of the 7 Baltimore, which are in the Baltimore classification for the this chapter. But you will see that as we go ahead in the course, you will be able to remember that what contain the DNA, what are the virus families contain the double-stranded DNA genome. We will use these over and over. So what are the variants of the DS genome? So this is the overall scheme that you can see that if you have a double-stranded DNA genome, it can make the mRNA. That will make the protein needed to make the box. The plus minus DNA will be replicated. This is the genome replication. So this is your mRNA synthesis. This is genome replication. This genome will be put inside the box made by the protein. And that's basically the overview of the scheme. And I will not go into the scheme because it's self-explanatory for the subsequent slides. But the main thing that what we are just pointing out, that what are the important things, that the first thing is that this DNA right here can be copied by the host DNA polymerase by these viruses like polyoma or papilloma viruses. And on the other hand, some of the other large viruses actually encode their own DNA polymerases. These actually steal the host DNA polymerase. They actually make their own. They do not steal the DNA polymerases. And then we have variant means that we can have circular double-stranded DNA. This is closest to your bacterial DNA, like an E. coli DNA is a closed circular DNA with 1 chromosome. They are closest to that. You can have that type in the polyoma papillomaviridi. And then again, you do not have to memorize each one of them, but you can have all the different ones. And the first one is that which have a 5' terminal phosphates attached to them. And then what you have, that you have this inverted terminal repeats that you can see that at the end, the ITR means inverted terminal repeats. That means you have a sequence, and on the opposite side, you have the inverted sequence going in there, and we will talk about that in detail. So you have the ends modified. On the herpes virus also, you have the L is the large and the small segment, unique large and unique small segment in the genome. These are very, 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 very complex because it makes a huge number of a frame, which makes the mRNAs and the proteins. And then TRL and the TRS. So these are the terminal repeats in the large and terminal repeat in small. Then IRL, IRS is internal repeats, large, internal repeats, small. And then ORI is basically the origin of replication. And L or S means that it is where in the segment. So this is your L segment. This is your US segment, large and small. But this is just a double-stranded DNA. It's just only single strand has been shown. But remember that it's a double-stranded DNA genome, which is not shown like this here. On the other hand, these pox viruses have internal term. This is the inverted terminal repeats at the end, but they actually have a very interesting structure. Their ends right here in these sequences, they are covalently joined. Here it is different. Here, see what happens. The same strand, the 3' end of the plus strand, is covalently attached to the 5' start of the 5' strand. So this is your intra-strand covalency. So the plus strand covalently joined with the plus strand end to end. The minus strand joins end to end. Here it is not like that. Here what happens that they are joined end to end, and as well they are joined covalently in these sequences as well. Rather than just base pairings, they will remain in this, and that keeps the genome integrity. So this is also very interesting structure. So here you can see that this is a gapped double-stranded DNA genome, and these are found in the reverse transcribing DNA viruses. And here you can easily see that a very complex type of genome, whereas you can see that the plus strand, which is the dark blue, is like this, and the 5' end of this strand does have an RNA in there. On the other hand, you can see that the light blue, that's the minus strand, the 5' end is attached with a protein. The yellow one is a protein as well, and there is an additional protein in there as well. So what they do, that it's found in the Hepatina viridis, and this was one of the class that Baltimore did not originally put in there because it was not discovered. So you have this gapped DNA. First, this gap is repaired so that there is a full length of the plus minus DNA. And from that, the mRNA is made, which makes the protein to make the shell or the box or the capsid. But this is the interesting part. So that from this DNA, plus minus DNA, what you will think that this is a plus minus DNA, and you can directly make the plus minus DNA from here to here. And this replication should be similar to any eukaryotic or prokaryotic replication, just double-stranded DNA to double-stranded DNA. This virus does not do that. And it, from this plus minus DNA, it makes a plus RNA, just like the mRNA. And from that RNA, it makes a minus DNA, negative sense DNA. And on this minus DNA, it makes this plus DNA, which is incomplete as well. And this step is the reverse transcription. And this particular virus actually do code for a reverse transcriptase. And why this is useful, or how this is useful for a viral, or why it is, well, sometimes in virology you cannot ask you cannot ask that question. You can ask is how. We can answer the process, but why we may not have any answer. That why this complicated. There was much easier solution, but why this evolved over the time, we don't have those answers. Then we are going into the single-stranded DNA genome. The single-stranded DNA genome could be plus or the minus strand. And whatever it is, it first needs to make the complementary copy so that it becomes a duplex DNA. And from that duplex DNA, you can easily see that it can either make the plus DNA on the minus strand or make the minus DNA on the plus strand. And of course, from this, it will get the mRNA to the protein, and either the plus DNA or minus DNA can be packaged inside the box and your whole virion. So classic example of this is a circoviridis. It's the smallest known virus to us. And this is a TT virus, and this is ubiquitous human virus. And the funny part is that each one of us will have this circoviridis in us, no matter what. It does not cause anything, but it is found in every human being, a very small virus. On the other hand, this parvoviridis has a very unique genome, and this is a kind of an illustration and why you have this T-shaped end. And we will see that these T-shaped end has something to do with the replication and transcription or mRNA synthesis of these genomes. And this parvoviridis is also very small, larger than this, but small, and it causes a fifth disease. And you can just read at the bottom that why it is called a fifth disease. It causes a rash in the children, and these rash diseases are basically the mumps, rubella, and all those. And these are the classic diseases of rash diseases. And this was the 1/5 in the list, and that's why it's called the fifth disease. It clears pretty easily. It's not lethal or anything of concern, but it still can cause a rash in children, particularly in the face, very prominently. All right, so then we are switching back to the RNA genome. Many varieties of the RNA genome. So you can easily see that the main concern is that the cells have no enzyme. This is the most important part, and this is the central to this understanding, have no enzyme, which is an RNA-dependent RNA polymerase. We will use this term RDRP for short. What we mean by that in any of the eukaryotic or prokaryotic cells, if you have a piece of RNA, you know, the mRNA, ribosomal RNA, or tRNA, or microRNAs, mini RNAs, whatever RNA you have in a cell, you cannot make an RNA copy from the RNA. It is always you always make the RNAs from a DNA copy in the organisms that we know of. So here you have a unique enzyme which is an RNA-dependent RNA polymerase, and this is needed for most, I say most, just one or two exceptions, to replicate their genome. And many of the times these are inside the virion, and these are not supplied by the host. And many of the RNA virus genome encode the RDRP, or RNA-dependent RNA polymerase, and they produce the RNA genome, mRNA, from the RNA templates, not a separate enzyme. The same enzyme, by a different mechanism, can make the mRNA as well as they can make the genome RNA as well. The only exception that we talk about is that hepatitis delta satellite actually can make the RNAs by reprogramming the RNA polymerase II that is found in the eukaryotes. And these RNA polymerase II is basically make RNAs from the DNAs, but the only exception is that the hepatitis delta satellite actually can reprogram this to make its own RNA from the RNA to RNA. So that is the only exception. So we're going to see that all the different ones with a single-stranded, negative-stranded, segmented, double-stranded, then circular, we're going to see all the different types of RNA genomes. So first, we are looking at the double-stranded RNA genomes. And in the double-stranded RNA genomes, you can see that there could be segmented as well, or it could be linear. So here, what we are seeing an example of a Reoviridae family, and they have a segmented double-stranded RNA genome. So you have 1, 2, 3, 4, 5, 6, 7. So you can see that there are many segments in right here. And it's not really, you cannot tell that these are like the chromosomes, but kind of an analogy, you can think that we have multiple chromosomes and there are different genes in each one of them. And this is the same thing for them as well. And they're large, middle, and the small segments in there. And they're all packaged inside the virus, double-stranded segmented RNA genome. On the human rotavirus is another example, which can also have the double-stranded genome as well. So it's pretty easy. From the double-stranded RNA, you can make the mRNA directly, which you can directly make as a copy on top of your minus strand. And from that, you can make the protein the same way. And again, for the making the double-stranded RNA, first you make the plus strand RNA from here, and then on this you make the minus strand, and that is packaged into the virus. So that's pretty much. And many of this, actually, the important part is that the process that actually works, particularly for these viruses, the RNA-dependent RNA polymerase is actually, so what happens that you can easily see that how is the process. Once these proteins are actually made, the mRNA is packaged into the virus as a single strand. And the RNA-dependent RNA polymerase for these Reoviridae family are also packaged inside the capsid or the box. And inside the capsids, these mRNAs actually are converted to the double-stranded RNA by the RNA-dependent RNA polymerase, which reside inside in there. So this is the important thing for that very unique strategy. So here we are looking at some of the plus RNA sense viruses. And this is the most viruses on this earth that is known actually belong to this plus sense RNA. So that's actually the interesting part. You can see some of the viruses that's known to you: picornaviridae, poliovirus, rhinovirus. Rhinovirus is the cause for the common cold. And then coronavirus, of course, all of them belong to this class, plus stranded RNA. Then you have a flaviviridae. And the flaviviridae actually do contain the yellow fever, West Nile, hepatitis C, and the Zika virus. Then you have a togaviridae, rubella, equine encephalitis virus of these families. And these are just the representative structures of them. And you can see right here that out of all these plus sense RNA, the coronavirus viridae family actually, and again, remember that the coronavirus family have that alpha, beta, gamma type of coronavirus, and the SARS-CoV-2 belong to the beta coronavirus group in there. There are many, many coronaviruses. And again, remember that there are many coronaviruses that infect human beings and cause cold as well. So, but they are just not SARS-CoV-2. They are different coronaviruses. So it is known for the human beings for a long time. This one is just different in the sequence and the virulence. And these one actually does have the largest genome size in all RNA viruses. As I said, that greater than 30 kilobase of genome size. And then beyond that, most of them are the DNA viruses. So here we are looking at the scheme of the plus sense, and you can see that the genome is plus sense, and this plus sense genome, you can remember that from your the slide 6, where we just wrote down the mRNA and the plus sense RNA, and why we separated it out. Many of the times, the genome can act as an mRNA itself, and this genome upon entry, so basically the virus goes into the cell and deliver the genome inside the cell, and that is translational ready. It can make the viral protein, which will make the capsid itself. On the other hand, the genome will make an RNA intermediate. On this RNA intermediate, it will make the plus strand, which will be packaged into the virus. And you will see that this is basically how it is described in most of the microbiology textbook. But there are some exceptions. Not all plus RNA genomes are mRNA. Means that some of the genomes upon infection need to be processed before they can make the mRNA. So not all plus-stranded. So do not take in the idea here, although in a microbiology course, this is okay, like 30, 50, this is okay. But here, just know that some of the plus-stranded RNA genomes are not mRNAs, need to be processed with that. So a couple of examples, again, you do not have to memorize all of that, but you can see right here, the coronaviridae with an untranslated region, and this actually tells you that this is the poly A tail of that. Then you have the flaviviridae family right here, which doesn't have a poly A tail. On the other hand, picornaviridae, which is a classic example of this, is a poliovirus, does have a VPG protein covalently attached to this region as well, and the toga virus, and some of them can actually directly be translated into a protein. Say, for example, the picornaviridae, which is a poliovirus, can actually make a full-length polyprotein out of this mRNA. What do we mean by the polyprotein? The same the full-length genome of that, or just coding region, the green region, it contains many genes one after another. And when this is read by the ribosome, they make proteins which are joined end to end. And this also codes for some proteases, and these proteases actually do chop this protein up to make those individual proteins that we're going to discuss later, not needed in here. So next we are looking at that the ssRNA plus sense with the DNA intermediate. And there is the one viral family, which is the retroviridae, which belong and commonly known as the retroviruses. So that is a classic example with that. Now, there could be as many as five orders of that, not families, orders of that. And some of these orders belong to the transposable retroviral elements that are put into that family that are found in our genome. So we do have a huge amount of retroviral sequences in our genome, and they are called the HERV, or the human endogenous retrovirus sequences. And there are few which can actually form virus-like particles in the embryos. And we will talk about that later in the course, but most of them are non-infectious or don't cause any harm. But some of them are regulatory in the sense they are absolutely needed for the gene expression, including pregnancy. Gene expression is controlled by these retro elements. We do not call them retroviruses because they are integrated in the genome. And as most of them do not make complete retroviruses, we call them retro elements rather than retroviruses. So classic example of your HIV. This is a cartoon of the HIV, which actually do contain 2 plus strand RNA. So you have 2 copies of that. And this is the interesting part of the HIV right here. Both are identical plus strand. Don't think that this is a plus strand, the other is a minus strand. This is the plus strand and this is the plus strand. Both are same polarity. Both are 5' to 3', exact same sequence. And this is one of the reasons that they just keep one as a backup copy. If one actually get destroyed in some process, they can actually switch the template to get it back from the other one. We're going to talk about those later. And these are some of the other proteins associated with the genome. Then human lymphotropic virus, HTLV. Any of the cat lovers, you know this very, very well. The feline leukemia virus, you actually get your cat vaccinated with that. But if you have adopted a cat or picked up a cat from somewhere, and before you actually give them the vaccine, usually the doctors or the vet actually do a quick feline leukemia viral test, because here is a problem. If the cat does have feline leukemia virus, which is, if they're positive, then the vaccine will not work. Because this is the important part, that all these retroviruses, upon infection, they will convert this RNAs, whatever it is, it could be single strand as well, the plus RNA into a minus DNA, then it will make a double-stranded DNA. This DNA will get integrated into the chromosome or the genetic material of the host for life. So once you have a retroviral infection, and that's the reason that if you have an HIV positive, you will be HIV positive all the time. Now, don't get me wrong here. What you can do, that even after that, you may not have viruses in your blood or the serum to make you sick, because you can take antiviral. So the virus that is being produced from those sequences may not be in your blood or the serum to kill the cells, the T cells that they kill, but the sequence in the genome will be there for your lifetime. You cannot get rid of it. So essentially what happens, some of these viruses actually are, which are integrated into the genome, are called proviruses. And these proviruses sometimes will just make the mRNAs replicate themselves to make a viral infection. Sometimes they don't. It depends upon several signals, and sometimes you can tell in a crude language that depends upon the mood of the virus whether they want to make viruses or not. But they are always in there. And all of these actually contain the reverse transcriptase, because these are plus RNA with a DNA intermediate, very, very unique virus. So here we are looking at the scheme of that whole thing. So you can see that this is the retroviral genome, and this is from this genome. And again, these use of the untranslated regions, does have poly-A tail as well, so plus-stranded RNAs. But from these plus-stranded RNAs, they make intermediate minus DNA, as I said, double-stranded DNA. And from this right here, the provirus, this is the stage at which the virus is integrated into the host chromosome. And it is always, this is the part. Once they're integrated in the chromosome, this DNA is a part of the chromosome, and when or where it is willing to, this DNA can make the plus RNA, which is the genome, or this can transcribe into the mRNAs, and this is using the host machinery. Here, they will use the RNA polymerases of the host and get splicing, make mRNAs, all the editing, and they will make the protein shell, and this will be packaged inside that. And this is basically the retroviral strategy. Very unique strategy, very different from every one of them. So after that, we are going into the minus sense RNA, ssRNA. And the interesting part is that the minus strand RNA, let's first go through some of the names. Then again, you do not have to memorize, but just keep in the back of your mind. You have the measles, mumps, rabies virus. Then you have the Ebola virus, Marburg virus, influenza virus, Lassa virus. These are all with that. I used to work with the Rhabdoviridis family, vesicular stomatitis virus, which is a representative of the rabies virus, which is a model. So just years ago, though. But again, the important thing is that this is the interesting part, that this negative sense RNA viruses are not the largest in terms of the families, like plus-strand RNA viruses. But this family actually do contain the deadliest viruses that is known to human beings. And interestingly, you can see that the Ebola virus, this Marburg virus, this Lassa virus, and these are all the viral hemorrhagic fever viruses with 80%, 90% mortality. Rabies virus, these are although we have vaccines, but these are, once you have that, it's almost impossible to survive from the rabies. Very, very dangerous. And that's the reason that most of the time in the literature or in the books or in the movies, whatever virus that's being portrayed are always actually comes from this family. It's kind of funny in there. So here is an interesting story. Now, remember that this book is not, this is a true story. This is not a fiction. There are lots of fiction out there based on that. But this is what happens, that this fever, this book, if you just had it long back, but if you just can read that, it will be great. So this Lassa fever virus was actually found in some of these nurses who were working in Africa and Nigeria, and they were flown back to the country. And they actually did survive. And then at the Yale School of Medicine, a group of scientists actually started to work on them. And what they did, they were working at that time. And unfortunately, if you can just go to that link itself, and you can just read the story about that, the PI actually got, or the principal investigator actually got really sick, and he was almost dying. And then he was actually given a plasma therapy or the serum from one of the nurses who actually survived the disease, and he recovered. Unfortunately, after that, one of the lab worker who was not in the lab that used to work with this virus, Lassa fever virus, but a different lab close by, a lab technician got this, and he did not survive. So what happened, that why I just put this story in there, before this is in 1969, when you're working with the viruses, the protocols of safety was not there. But once this happened in 1969, 1970s, there was an enormous stringency in the protocols that how you can handle these agents, and then we can have these different strains, and they are handled under specific biosafety level, like a BSL 1 through 4 is a common biosafety level, BSL 3, BSL 4, like if you just had to handle Ebola, Marburg, Lassa fever virus, you need a BSL 4 facility. The SARS are just handled into the BSL 4 facility, but I don't think that at that time the scientists are pushing that it is not needed to work under a BSL 4 facility, and it will probably be allowed to as a BSL 3 lab as well. Now, why I said that, because BSL 4 facilities, there are just a handful of few throughout the United States, and you just need to wear basically a spacesuit kind of kind of gown, and have air supply. So there's an air supply line at the top, and you just hook it up from the back of your suit, and you just have to work in a 100% contained environment. And that facility is very difficult to maintain. There's a newest one in the Boston school. Boston University had the latest one in there. There's one in Galveston, and there are a couple more in there as well, but not many. All right, so let's go to the real thing. The negative strand RNA sense viruses, on the other hand, you have the negative sense, and to make the RNA, it just have to copy the complementary plus strand, which will make the protein, and that will make the shell of the virus. On the other hand, the minus strand are converted into the plus RNA. From there, you get back the minus RNA that will be packaged inside the genome. Now, the important is that the orthomyxoviridis family actually do have minus segments, and these are segmented. This is just like, if you remember the reovirus that we talked about, which is a double-stranded RNA virus. They have segmented genome, but each of their genome is double-stranded. Here it is similar, but these are all minus-stranded. And a classic example of this is your influenza virus. Your influenza virus have 8 of these segments, and they belong to the orthomyxoviridis family. On the other hand, you have a non-segmented genome as well. They are negative sense, like a paramyxoviridae, and this is a human parainfluenza virus, is belong to that. Rhabdoviridae, which is a rabies virus, belong to that. They're just only one straight single negative strand. So we are going to talk about that this segmented RNAs in this particularly, this is a model of the influenza virus that you are seeing right here. We will use this and what you're seeing that these are the hemagglutinin and the neuraminidase proteins on their surface that you are seeing. This actually gives rise to a very interesting phenomenon that we're going to talk about in a second. So here, what happened here? This is a duplicate slide. Sorry about that. I will, somehow I did not delete this one, so this is deleted. Okay, all right. Sorry about that. Are we good with the sharing? Can you see the slide? I took it off. Okay, all right, thanks. All right, so yeah, we went over this one. So here is a phenomena that is very interesting. And what you are seeing, that is the same thing. These are the negative stranded segmented genome of the influenza viruses. And due to this, they can actually do a remarkable thing. And this is known as the reassortment. And this is basically, in a crude way, you hear this H and ends with a number, and all these different H1N1, H5N1, H2N2, H3N2, all and so on. And these combinations are possible due to this reassortments. So here is an example of an experiment that how you do that. So here, suppose a cell, and it doesn't happen to every cell, but you have a virus which is an L strain. This has a virus which is an M strain, and you can see that all of their genome segments, negative strand genome segments, are marked as a red. These are all blues. Now, suppose in a host, some cell got infected or co-infected with both of them. And again, remember that can happen. If you remember that we talked about, if one infects one cell, you will get a one-hit kinetic. If there is more than one, you can get a two-hit kinetic. And the question was asked in the plaque, if really one virus goes into one cell, cause that? No, it can be multiple. And that's the reason that we've also talked about that we purify the plaques as well. So when this happens in the cell, as these segments are being made, they can be packaged wrongly. So from here, the progeny, this is perfectly fine. This is perfectly fine. But see this recombinant 3 segments where the second segment of the M, the second segment comes from the L segment. That's why this is red. And this is happened with the hemagglutinin and neuraminidase combinations all the time, which gives rise to the different strains of the influenza virus. And this actually cause more problem because some of them are more virulent than the other. And this randomly happens all the time. And sometimes what happens that the scientists actually look at the viral strain that are being circulated into the environment, make the vaccine for that. But sometimes if this reassortment actually happened later in the season, you have a new strain, which is virulent as well, being circulated, and the vaccine will not work. This happens, I think around 2016 or 17, the vaccine was not that great, and this can sometimes happen as well. All right, so the last type that what we are talking about is called the ambisense RNA genome. That ambisense in the sense that you find them in the arena viridae and bunia viridae. So this actually do have 2 segments. And again, we're going to talk about that later, just an overview of the strategy. That's one part of the genome. This is the plus strand right here. And this part of the genome is the minus strand. And why you will call that this one is the minus strand, why you do not call that the whole thing is the plus strand. The reason for that is that the polarity of the genes in this is in a certain way that when they will be making mRNA, they will be particularly the way it is, the gene segments, that they will be converted into the plus segments, and from there you make the mRNAs. So the structure is basically tell them that these are the ambisense genomes as well. So that's the important part, and that is the reason that we call them plus or minus RNA, because see that some of the part can be translated, and then some of the part need to make the plus strand mRNA to get get the protein done or made. So the whole plus strand, this will not work as a whole plus strand RNA. If it is, then it will get translated from this end to this end. But what happens that only this part gets translated into the protein, and then this part need to make a plus strand, and that part actually is translated. So all these weird combinations, and again, details are not needed here. Just you know this, and we will go to talk about this later. Now, the extensive stem loop structure that you can see that the genomes are not like that, how we draw them, but they have extensive stem loop structures, secondary, tertiary structure with long distance interactions. Now, the last part that we are going to talk about in 3 minutes, that we can do a huge amount of mutations into the viral genome into the modern way. And we are not covering the last part in the sense that as we are not working with the hand, we're not going into the detail of the experiment. But we can clone all the viral genes into a plasmid in the bacteria, and you can transfect, means put it in an animal cell, and you will have a virus that actually comes out. We can engineer a lot of different things, but we cannot engineer a lot of different things. And we'll argue about this. I just put that in the notes that you can read, very self-explanatory. And we are going to argue about the source later in the chapter. So here, the scientists were able to resurrect the 1918 influenza virus. They actually got these from the frozen samples of the in situ biopsy of the lungs. They were buried in Alaska in the 1980s from the permafrost, and also isolated from the paraffin-embedded tissue samples of autopsy victims. And the complete sequence of all 8 RNA segments of this H1N1 is obtained from that as well. And sometimes you may not have baked this infectious clone into the plasmid to get a virus out. Nowadays, you can just chemically synthesize the entire genome like this in the horsepox virus that has been done, and you can just transfect that into the cells to get an infectious virus out. So you can do a lot in there, and we'll go to talk somewhere else in there. And that brings that all these biosafety, synthetic virology related biosecurity questions about the ethics, what it is. And the main thing that is important is that this actually caused a huge amount of stir outside the scientific community. And as we can do a lot, a huge amount of conspiracy theories will go out. And sometimes even the scientists will question that, is it true or is it not? But what happens that most of the times, looking at a viral sequence, there is a signature in there that how it evolved. You have a lineage of that. And that gives you the insight to bust all the myths or the conspiracy theories that go around. The science is at least that smart at this point of time. Some of them, we should not boast ourselves that we know everything, but at this point, we do think we know that what is going on from the scientific background. So that's about it.